Set of lectures in histology, and today's topic is blood formation. So it is one of the toughest topics in this semester for you. So please please be attentive in this lecture. So the record of this lecture will be present in YouTube channel, and I'll send it in Google Classroom and personally to groups in WhatsApp. Okay, let's start. So 1 minute. Yeah, let's start. So blood formation. So another name is hemopoiesis. Hemopoiesis. So one second. So hemopoiesis, it's blood formation. So or hematopoiesis. It's not mistake to name both hemopoiesis and hematopoiesis. It includes erythropoiesis, formation of erythrocytes, which we discussed in previous lecture, and leukopoiesis. It's formation of leukocytes and development of red and white blood cells. So together is known as hemopoiesis. Includes erythropoiesis, leukopoiesis, and also thrombopoiesis, formation of thrombs or platelets. Blood cells have limited lifespan, so why do we need to produce blood cells every single day? So every single day produced a numerous amount of the cells. You remember that lifespan of every cell of blood is limited. For example, for erythrocytes it's 120 days. For leukocytes, for different leukocytes, it depends on type. And platelets life is approximately 10 days, and we need to reproduce them every single day, so in numerous amounts. And it's not a secret for you that blood formation goes in bone marrow, in a red bone marrow. So later in topic of bones, we'll discuss the location of red bone marrow, but you understand that red bone marrow present inside the bones and in special locations of the bones. And later I think in anatomy, you will discuss this too. And in histology also we'll continue the discussion. Okay, and in adults, erythrocytes, granulocytes, monocytes, and platelets, they are formed in red bone marrow. Lymphocytes also formed in red bone marrow and in lymphatic tissues. So how it goes, let's discuss. So, but hemopoiesis initiated in early embryonic development. So in previous semester we discussed the embryology, and you remember that during the embryogenesis we have a stage of yolk sac. So we have yolk sac, it's prominent, and the size of yolk sac is even larger than entire embryoblast. So larger than the rest of embryoblast, and but gradually yolk sac shrinks and enter into the body. So first blood formation initiated in yolk sac phase. But later, this from yolk sac phase, blood formation is shared to hepatic phase. When yolk sac is shrinked, and liver develops, so all the hemopoietic cells entered into the liver, and this is known as hepatic phase. And later, when fetus is developed more and more, so we can find blood formation cells in a bone marrow. And also this thing I'll try to show it to you in diagram here. So this, you remember this embryo. So this is trophoblast around, and this is embryoblast. And embryoblast is composed of amnion, yolk sac, and mesoderm between them. Yeah, so I'll try to draw it. So this is amnion, this is yolk sac. Between them we have mesoderm. So around we have trophoblast, so all components of trophoblast, like blastocyst or this extraembryonic mesoderm, or this cytotrophoblast and syncytiotrophoblast. But now our attention is concentrated on this yolk sac. So actually formation, so yolk sac is made up of endoderm. remind you, endoderm, and but near the endoderm we have this extracellular mesoderm. So this extracellular mesoderm, visceral extracellular mesoderm, and inside this extracellular mesoderm formed the structure which is known as blood islets. Blood islets. So let me draw it. So, mesoderm is related to the spindle-shaped cells, and they are present in chaotic arrangement. This way, in chaotic arrangement. But so now let me join one student and I'll continue. Okay, in chaotic arrangement, but gradually in yolk sac, due to presence of some specific factors, these cells start to arrange this structure. This structure like this. So this is nucleus. There are nucleuses, and inside, and some of these mesenchymal cells enveloped inside here, and they are free, non-attached. And you remember, yeah, that these cells became tightly attached to each other, and they forming something the epithelial tissue arrangement. So like epithelial tissue arrangement. And if you remember the epithelial tissue topic, you can explain me what's the name of this epithelial cells. And the name of this epithelial cells is simple squamous epithelium. It is formed from mesenchymal cells here. So these mesenchymal cells start to join each other and form the simple squamous epithelium, and form something like bubbles inside the yolk sac, inside the mesoderm of yolk sac. This is known as blood island. So, okay, blood island. Blood islands. And these blood islands are it's a predisposure for future blood vessels. So this simple squamous epithelium later will be present in blood vessels. So as we discussed previously, and but now they are just like bubbles. But later these bubbles inside the yolk sac, they start to join each other and forming the capillary network. You know what is capillary, yeah? And they join together, and this is primitive blood vessels are formed. But cells which are present inside these cells, they form the HCT, HST, sorry. HST is hemopoietic stem cells. HSC, sorry, hemopoietic stem cells. These hemopoietic stem cells later will form the oolenix of blood population. So they are primordium germ cells, primordium stem cells for the blood formation. Okay. And then, later, this pattern of blood vessels from yolk sac entered inside the mesoderm. So when continuation of development of embryo goes, so these blood vessels entered into the embryo, and now inside embryo we have blood system. So it's oversimplified point of explanation of blood vessel formation, but it's not the charge of our explanation right now. For us, more important is these cells. These hemopoietic stem cells, and these hemopoietic stem cells now entered inside the embryo, and then inside the embryo produced the liver. So it's evolved from endoderm and mesoderm together, and these hemopoietic stem cells start to attach to the liver organ, and liver now is the main organ of hemopiesis. This is the hepatic phase of hemopiesis. But later, when embryo became not embryo, but when embryo became fetus, so more resembling the baby, yeah, liver still facilitate functional blood formation, but along with it, bones are formed. So I remind you that bones are based on cartilage. So here we see cartilage, and gradually this cartilages became ossified. Ossification includes the calcification of surface and the growth of osteoblasts, and then the formation of osteocytes, which we'll discuss a little later. And but more important for us is that inside this bone entered the blood vessels. Once again, blood vessels now carrying the hematopoietic stem cells. Hematopoietic stem cells are present right now, and along with these blood vessels in the bone entered these blood-forming cells. So we have primary ossification centers. This is primary ossification, and then this is secondary ossification center. But anyway, inside the bone appeared a very pronounced network of blood vessels. So. And that's how gradually from the liver it enters into the bone marrow, and that's known as bone marrow phase. Once again, now you understand the point. First of all, we have yolk sac phase when development goes in yolk sac. Then we have hepatic phase when gradually this hematopoietic stem cells entered into the liver, and finally it reached the bone marrow phase. So when most of cells entered into the bone marrow. But point is that transmission goes differently. So we can say the date when yolk sac phase continues in liver hepatic phase, and then when hepatic phase is changed into the bone marrow phase. It goes approximately this way. So let me draw it this way. So let's take a look at yolk sac phase is this diagram. So in beginning of embryogenesis, all the blood formation that happens inside the yolk sac. This is the yolk sac phase. So okay, then minute. Then and approximately in the middle of this yolk sac phase appeared the hepatic phase. So when most of blood cells are produced here in hepatic phase, and gradually when bones are appeared in the body, so starts the bone marrow phase. And bone marrow phase exists until the mature period after the delivery and until the end of human's life. And so blood formation goes in bone marrow. So that's why we can say the dates, exact dates when this phase is over and this phase starts. But approximately we can say that yolk sac phase present only until the yolk sac phase present only until the end of embryonic period when yolk sac shrinks. Then hepatic phase present only until the birth, and bone marrow phase initiated in fetal period and continued after the birth. And usually we use this timelines. So this 8 weeks. Until 8 weeks we have yolk sac phase, until birth we have hepatic phase, and after birth we have bone marrow phase. It's approximately, but keep in mind that they are overlapping, these periods are overlapping. Okay, then let's switch to monophyletic theory of hemopoiesis. Monophyletic theory of hemopoiesis is a theory which posted that which established the understanding of this. Monophyletic theory says that we have only one cell. We have this hematopoietic stem cell, which finally forms the all cells of blood. So all cells of blood which we discussed in previous class, they are derived from one cell, hematopoietic stem cell, and this position is of monophyletic theory. That's main theory. But these hematopoietic stem cells later get differentiated by the process, by the diagram I'll show you later, Okay, another name of hematopoietic stem cell is known as pluripotential stem cell. And this pluripotential means that this cell is able to produce a lot of different cells. Pluripotential means a lot of new cells, because we discussed in embryology we have totipotential stem cells which are responsible only for all cells of human body formation, but pluripotential is responsibility is only for blood cells formation. Okay. Hematopoietic stem cells in the bone marrow gives rise to multiple colonies of progenitor stem cells. So, and we have two main progenitor cells. We have two main progenitors. They are common myeloid progenitor, so common myeloid progenitor, and common lymphoid progenitor. Common lymphoid progenitor. This first division of hematopoietic stem cell. Hematopoietic stem cell splits into these two types. In diagram, I'll show it to you again. And common, and let's consume. Yeah, so common lymphoid progenitor is responsible for all lymphocytes formation. But common myeloid progenitor is responsible for all other lineages of formation. It includes red blood cells, platelets, and granulocytes. Yeah, all of them are produced in common myeloid progenitor. So please keep it in mind, because usually students have confusion here. So that if we have, so usually you think about this like this: leukocytopoiesis, erythropoiesis, and thrombopoiesis. But actually, erythropoiesis and most of leukocytes and thrombocytes, they're derived from one cell. It's a common myeloid progenitor. And only lymphocytes, they possess their own branch of cell formations, common lymphoid progenitor. And their differentiation goes inside the bone marrow. Okay, let's talk about common myeloid progenitor. Common myeloid progenitor, so that's myelin. It's also known as a CFU-GEMM. CFU-GEMM, which is translated like colony forming unit. So CFU. So if you see CFU, it's known as colony forming unit. Now, colony forming units, it's for granulocytes, erythrocytes, monocytes, and megakaryocytes. So that's why it's GEMM. Granulocytes, erythrocytes, monocytes, and megakaryocytes. And this what means colony forming unit? So must be these cells are able to form colony inside the liver and later inside the bone marrow, and this colony will be responsible for formation of more and more cells. These cells are like settling in these organs, and you can and they can continue their differentiation and blood formation. And later, the CFU-GEMM is differentiated in lineage-specific progenitors, which are known as MEP, megakaryocyte erythrocyte progenitor. So, according to name, you can conclude that it's responsible for erythrocyte formation, and megakaryocyte. What is megakaryocyte? We'll discuss a little later, but a little spoiler: it's responsible for platelets formation. Platelets. And another progenitor is also known as GMP, or another name is CFU-GM, colony forming unit, granulocytes, and monocytes. GMP, granulocyte monocyte progenitor, responsible for formation of granulocytes, neutrophils, eosinophils, and basophils, and monocyte, which is monocyte actually. Okay, let's another lineage of cells, which is known as common lymphoid progenitor. Common lymphoid progenitor cells are responsible for formation of leukocytes, and there are three types of leukocytes. We discussed it before. It's T cells, B cells, and NK cells, natural killer cells, and they produce their own respective progenitor: T cell progenitor, B cell progenitor, and NK cell progenitor. So let's talk about the erythropoiesis. Let me show you a general diagram of blood formation, then we'll discuss it. So here, this diagram, so now might be, if you use smartphone, it might be hardly visible for you, but try to understand. So this is hematopoietic stem cell. This is the first cell of blood formation, which was enveloped in blood islands, as I said before, in yolk sac. This is typical mesenchymal cells. It was mesenchymal stem cell. And this mesenchymal stem cell later during the development is differentiated into two cell lineages, as I shown you before. It's a common lymphoid progenitor, which is responsible later to formation of lymphocytes and K cells, T cells, and B cells. So this plasma cell we'll discuss a little later. And another lineage is responsible for it's known as myeloid branch of hematopoiesis. It produces common myeloid progenitor, CFU-GEMM, and CFU-GEMM later differentiates into two lineages. It's megakaryocyte erythrocyte progenitor and granulocyte monocyte progenitor, which is responsible for both granulocytes and monocyte formation. Okay, and now we'll concentrate our attention on this branch. Of hematopoiesis, erythropoiesis, which is usually the most important for us type of formation because of diseases known as anemia. They are predominantly related to this lineage of blood formation. Let's return to this diagram. Yeah, erythrocytes. So this is general plan of development. First of all, we have megakaryocyte erythrocyte progenitor, which furtherly splits into megakaryocyte progenitor cell and erythrocyte progenitor cell. Erythrocyte progenitor later gradually collect more hemoglobin inside the cells, then nucleus extruded inside the bone marrow, and then only when mature erythrocytes is formed, it released inside the blood vessels. In previous lecture, someone asked me about this. Yeah, that Muzamil Siddiqui, I thought, asked me about this. That nucleus extruded inside the bone marrow, not inside the blood. In the blood, we can find only unnucleated erythrocyte. So, but let's talk about more details about it. Here, this lecture is uploaded into the Google Classroom. That's why I'll stop only on diagrams, and all the necessary information about blood formation you can find in this PDF lecture PDF. So take a look. Proerythroblast. It is usual cell with a very huge nucleus. Inside this nucleus is absolutely usual DNA content, so like in every cell. But this proerythroblast inside the nucleus here produced a lot of nucleolus. So nucleolus are located inside the nucleus, and as you remember, nucleolus function is formation of ribosomes. So that's the only difference of proerythroblast from other stem cells. They possess a lot of nucleolus, and which are responsible for ribosome formation. And in the next step, cytoplasm. In cytoplasm found a lot of ribosomes. Yeah, these ribosomes, and they are shown in histology like blue cytoplasm. Why is it blue? Because ribosomes contain RNA, ribonucleic acid, and acidic components of the cytoplasm will attract more basic stains. Basic stains like hematoxylin or methylene blue. Both of them blue in color, and both of them will color the cytoplasm of this basophilic erythroblast into the blue. That's why it's called as basophilic erythroblast. So now keep in mind that basophilic erythroblast is basophilic due to presence of ribosomes in their cytoplasm. And you can understand that what's the function of ribosomes is the production of hemoglobin, because erythrocyte must possess hemoglobin in the end. So and that's why. So still someone joining. Ainat Zahra. So and later and you can understand that these ribosomes require any RNA. So they require messenger RNAs from DNA to produce hemoglobin. One more point. These ribosomes are free ribosomes. They are not related to rough endoplasmic reticulum. These ribosomes are free because they need to produce their product inside the cytoplasm. We need to collect this hemoglobin inside the cytoplasm. So this is basophilic erythroblast. Once again, proerythroblast produce create a lot of nucleolus, and this nucleolus they will produce ribosomes. And when ribosome amount is very high, so we can name it like basophilic erythroblast because of basophility of their cytoplasm. Later, basophilic erythroblast, ribosomes of this basophilic erythroblast start to produce hemoglobin, and hemoglobin is acidophilic or eosinophilic. So, and it will attach eosin stains. And that's why here in this cell, in polychromatophilic erythroblast, we can find both stains, and acidophilic and eosinophilic. Eosinophilic and basophilic stains. And that's why color here is changed. Color here will be mixed up. And this mixing of colors here named as polychromatophilic erythroblast. It is named as polychromatophilic erythroblast. This polychromatophilic erythroblast is so you see, when amount of hemoglobin and amount of ribosomes is in equilibrium, and nucleus here is still quite large. So because of what? Because nucleus is still active, because euchromatin of this nucleus producing mRNA for hemoglobin synthesis, which is required for hemoglobin synthesis. And still inside the cytoplasm we have a lot of different organelles like mitochondria, Golgi apparatus, endoplasmic reticulum, and so on, lysosomes, all the stuff we discussed in cytology. They are still present in this cell. But later, this polychromatophilic erythroblast collect more and more hemoglobin, and larger condensation of this hemoglobin inside the cell, so causing the more acidophility of the cell. And that's why this cell is sometimes named as orthochromatophilic erythroblast, like in this book, where I get this diagram, or another name is acidophilic erythroblast. Orthochromatophilic or acidophilic erythroblast. 1 minute. Okay, let's continue. Then orthochromatophilic erythroblast. What's the difference? Much more hemoglobin is collected, and until this moment, cells are dividing. Okay, and then orthochromatophilic erythroblast contain a lot of hemoglobin inside, and ribosomes are I said about this. So here actually it's shown like a transition from one cell to other cells, but actually it is followed with cell division, and here formed two basophilic erythroblasts. Then basophilic erythroblasts keep dividing, and then formed two polychromatophilic erythroblasts. Yeah, so another cell is formed here. So you see multiplication of cells amount happens. And polychromatophilic erythroblast is able for cell division. It also forms one more orthochromatophilic erythroblast. Yeah, this also forms two. This forms two. And polychromatophilic erythroblast is still able for cell division. But orthochromatophilic erythroblast is enable of cell division. Because of what? Because nucleus in the cell is shrinked. And all the genetic material here became very condensed. And you remember that in cell cycle we should have open chromatin. We should use euchromatin for cell division. But here cell division is unavailable because nucleus is shrinked. And actually cytoplasm is now unable for cell division, and that's why after this we don't have cell division. After orthochromatophilic erythroblast, cell division is dumped. And next process which is present here is extrusion of nucleus. Nucleus is extruded from orthochromatophilic erythroblast, and remained only cytoplasm of the cell. And cytoplasm with hemoglobin inside, and also some remnants of ribosomes inside, remnants of some organelles inside. They're still present inside this, and that's why it possesses some additional colors. Yeah, it is not like typical red blood cell, but inside we have remnants of other organelles, and that's why we named it as reticulocyte. But on the level of reticulocyte, these cells are able to enter into the bloodstream. bloodstream. They're able to enter into the bloodstream, and final maturation of erythrocytes goes inside the bloodstream. So I answered to a question in previous lecture once again. So nucleus extrusion goes inside the bone marrow, but final maturation of erythrocytes goes in a bloodstream. And then when all these remnants of organelles are destroyed, gradually destroyed, so all ribosomes are destroyed, all membranous compounds are destroyed, and here present only erythrocytes. Here present only erythrocytes in bloodstream. But in cases of acute blood loss, so we can find 1 to till the 4% of reticulocytes. So it used as clinical feature of blood loss or any kind of anemias. In anemias, so then in pathology we'll discuss how is it related. But in anemias, we have larger amount of polychromatophilic erythrocytes along the decreased amount of erythrocyte. So I hope it's understandable. Once again, from beginning I will say to you, we have proerythroblast with a lot of nucleolus which are responsible for ribosome formation. Then ribosomes are formed inside the cytoplasm of basophilic erythroblasts. The cells still able for cell division. Nucleus is basophilic due to ribosomes present there. Then ribosomes producing the hemoglobin inside the cytoplasm, and that's why cytoplasm became double colored. And double color is reflected in the name is polychromatophilic. Poly means a lot, multiple amount, and chroma is color, and then two colors is named as many colors. So is a colored erythroblast. So it's multicolored erythroblast, polychromatophilic erythroblast. Then gradually this amount of hemoglobin became larger than amount of ribosomes and formed the orthochromatophilic erythroblasts, or also named as or normoblasts. And they are still present inside the bone marrow. This is bone marrow. But when extrusion of nucleus happened, so nucleus just run away, and then later this nucleus will be digested by macrophages inside the bone marrow. But remnant of cell without nucleus will be named as polychromatophilic erythrocyte, or more commonly you can find the name reticulocyte. And then reticulocyte final maturation goes inside the bloodstream, and we have and later forms the erythrocyte. So about information about erythrocyte you can find in the lecture before. So and then let's talk about hemoglobin recycling. So you know that inside the erythrocytes we have hemoglobin, which possess iron also inside, and how we deal with it. So a lifespan of erythrocyte is 120 days. So must be we should produce every 120 day a new cell instead of a died cell. So that's why every single day 1% of died erythrocytes in the human body is replaced by newly produced erythrocytes that maintain the amount of erythrocytes in a body. But we said that 120 days. What happened with erythrocyte after the 120 days? This erythrocyte get destroyed by macrophages inside our body. We have macrophages which swallow this senescent erythrocytes, the old erythrocytes with damaged cell membrane or somehow so any damage to erythrocytes when it happened. So phagocytes will degrade these erythrocytes. Usually it happens in a spleen. That's why might be you remember in some classes you might hear that spleen is a graveyard of RBC. Graveyard of RBC. And this degradation of erythrocytes goes in that way. So hemoglobin, you remember the hemoglobin is composed of porphyrin ring with iron in the middle, with iron +2 in the middle, and attached to these proteins, alpha globin and beta globin. Yeah, and this globin is a usual protein, and this protein will be destroyed, detached from the hemoglobin, and destroyed until peptides and then amino acids. Amino acids. Amino acids are produced so after this globin degradation, but and that's it has no issue for us, yeah, and we just reproduce it. But what about iron? So we can't remove iron from our body. So iron should be maintained on the same level inside the human body. So please, Ayub Khan, switch off your microphone. Thank you. And this iron is carried by with the help of proteins known as ferritin. So ferritin along with iron, they are stored usually in spleen, but sometimes in excessive amount of this ferritin they might enter into the lungs and other parenchymal organs. But normally they are present. This ferritin is present only inside the blood or in spleen, and it is like pool for iron for the later use in formation of more erythrocytes. Another name of this ferritin is hemosiderin. Okay, but what happens to this frame of the iron? This frame of the iron is known as porphyrin ring. Porphyrin ring. Shamsatar joined. Porphyrin ring. This porphyrin ring is this porphyrin ring is very dangerous for our body. It's also known as cyancobalamin or porphyrin ring, and this excessive amount of this porphyrin rings inside the blood causing the liver degradation. It happened in some diseases like porphyria. Might be heard about porphyria. So, but we need degrade this porphyrin rings, yeah, and it happens in this in biochemistry. They'll explain it much better, but I'll try to show it to you. Okay, so it works this way. Okay, and this square-like porphyrin ring changed into this type of porphyrin ring. The disruption in this area, disruption of this square in this area, causing the formation of biliverdin. Biliverdin. So this is shown here or not? No, it's not shown here. So this biliverdin, and later this biliverdin is changing their shape to form finally bilirubin. Bilirubin. And bilirubin, you know this bilirubin from physiology, you discussed it. So bilirubin is the yellow colored pigment in our body, so and which is later degraded in liver and later forms and it gives the color to our fecal masses, it gives the color to our plasma, as I said in previous lecture, and it also gives a color to our urine. So this bilirubin is our pigment which explains a lot of changes in color in clinic. So, but it's not point. Pathology is not point of our today's discussion. But is it normal degradation of hemoglobin? Next point is development of thrombocytes, thrombopoiesis. So, and you remember that I said megakaryocyte erythrocyte progenitor. Yeah, megakaryocyte erythrocyte progenitor responsible for formation of both erythrocytes and platelets. And megakaryocyte is like a progenitor cell for thrombocytes. But I said to you that platelets or thrombocytes, they're not actually cells. They are just fractures of cells. In previous lecture I said about it. So let's talk about this, how it goes. Megakaryocyte erythrocyte progenitor gives rise to megakaryocyte progenitor, or another name CFU-MEG, a colony forming unit for megakaryocytes. Then megakaryocyte progenitor, after few cell divisions, forms the megakaryoblast. And what's the difference between megakaryocyte progenitor and megakaryoblast? Megakaryocyte progenitor is able for normal mitotic division. So it produces one more megakaryoblast, and yeah, it can produce one more megakaryoblast. But megakaryoblast is enabled for one more mitotic division. So instead of a mitotic division, here goes the process which is known as endomitosis. So endomitosis. Sorry, mitosis. Endomitosis. So endomitosis is a process by which we also have split. So we have a cell cycle in which produced double strands of DNA, and then these double strands of DNA also detach from each other in form of chromosomes. But it is this endomitosis, is a type of mitosis which is not followed with cytokinesis. So, and this after this we can see this enormously huge nucleus. This is nucleus of this megakaryocyte. So sorry for rough drawing. So, but it's tough to do it with the mouse. And you see extremely large nucleus which possess until 32 to 36. 32 for 64. Sorry, 64 amounts of chromosomes. So in normal cell, I remind you. So in normal cell, we have 2n chromosome composition. But in this cell, megakaryocyte, it's possible for 64n chromosome composition. And one cell, and this process is known as polyploidia. Polyploidia. So in the text, you can write the spelling of this name. And in cases of polyploidia, cells are growing in size, and cytoplasm is enormously large. And that's why sometimes megakaryocyte is named as giant cell of bone marrow. And you can easily find it. It's the largest cell of human body. It's one of the largest cells of human body. Let's be correct. And actually, megakaryocyte later just split a small fractures of their cytoplasm to form platelets, and they extruded from the cell and form just fractures of cytoplasm. So that's why platelets can never have nucleus. Platelets, they are not cells, they're just fractures of cells, fractures of megakaryocyte. And megakaryocyte is always located only in bone marrow. But platelets which are produced here, they are directly added into the bloodstream. I hope you got it. Okay, once again, we have megakaryocyte erythrocyte progenitor. Differentiation of this goes to formation megakaryocyte progenitor. The megakaryocyte progenitor, by mitotic division, produce a lot of megakaryoblast. And these megakaryoblast, which they are unable for cell division, and they start to undergo the process known as endomitosis, by which nucleus became extremely huge, and it possesses 64 number of chromosomes, so 64 numbers of chromosomes. So you see one n is equal to 23 chromosomes, yeah? And you can multiply it, 64 numbers multiplied to 23 numbers of chromosomes. So, and that will be amount of chromosomes inside this nucleus. And this is enormously huge cell, enormously large cell, outpoach the small portions of their cytoplasm to form platelets. So this process of platelets formation, so it's a tip for you. It resembles the process similar in glands. You remember in glands, I explained you that we have merocrine, apocrine, and holocrine secretion. So this type of, if it's not secretion, but by process, by mechanism, it's similar to apocrine cell secretion. Apocrine secretion, so like portions of cytoplasm get outpoached. So if you remember the topic about glands, so you can find it that platelets formation is almost similar process. Okay. And then let's talk about the granulocytes, granulopoiesis. Okay, from myeloid progenitor formed another link. So this is hematopoietic stem cells here. It gives rise to granulocyte monocyte progenitor, and another cell is megakaryocyte erythrocyte progenitor. So megakaryocyte erythrocyte progenitor has been discussed now. So thrombocytes and platelets and erythrocytes formation we already discussed. But now we'll talk about granulocyte monocyte progenitor formation. So, and this granulocyte monocyte progenitor get differentiated first of all into the neutrophil progenitor, basophil or mast cell progenitor, eosinophil cell progenitor, and also, so here it's shown prodendritic cell, and also additionally, so here it's did not fit here, it is monocyte progenitor, MP, monocyte progenitor, and later it forms the monocyte. So let's talk about details. So let's talk about granulocyte formation. And then myeloblast, which is megakaryocyte, myeloblast is a cell so it is. So once again, I'll show it to you. Myeloblast. No, here. Okay, let's talk this. Myeloblast is here. So let's name this, but actually it's not. But okay, myeloblast is ancestry of these cells. But later myeloblasts are differentiated into promyelocyte. This is promyelocyte. This promyelocyte is furtherly divided into three types of cells. We have neutrophilic myelocyte, eosinophilic myelocyte, and basophilic myelocyte. And you can conclude, yes, that if we have promyelocyte here, how we can detect this? That they are different cells by possession of granules. In neutrophilic myelocyte, we can find polychromatophilic inclusions, granules. In eosinophilic myelocyte, we can find more eosinophilic granules. In basophilic myelocyte, we can find more basophilic granules. But take a look, nucleuses are still round in shape, round to oval in shape. But then next step of maturation is related to curving of the nucleus in the form of bean-shaped nucleus. So this bean-shaped nucleus and the possession of neutrophilic granules, it gives the name of neutrophilic metamyelocyte. Same bean-shaped nucleus and eosinophilic granules inside, it is known as eosinophilic metamyelocyte. And possession of bean-shaped nucleus and basophilic granules is known as basophilic metamyelocyte. Later, neutrophilic metamyelocyte, so nucleus of neutrophilic metamyelocyte became more elongated. It doesn't resemble the bean now, but it more resembles the stick, curving stick. Yeah, and it is called as bent neutrophil. Actually, we have the same stage in eosinophilic metamyelocyte and basophilic metamyelocyte, but we can detect them in blood, so because they are not so outnumbered. So who is trying to join closer to the end of lecture? Yes. Okay. Then this bent neutrophil. So final maturation steps going inside the. So yeah, once again. Until this moment, everything is present inside the bone marrow. But on the stage of bent neutrophil, so it starts the blood stream. It was bone marrow, it is bloodstream. And bent neutrophils, bent eosinophils and bent basophils, they're present in blood. And the final maturation happens inside the blood and forms a neutrophil, matured eosinophil, and matured basophil. Once again, sequence of events. Myeloblast produces promyelocyte. Promyelocyte produces three types of granulocyte myelocytes. We have neutrophilic myelocyte, eosinophilic myelocyte, and basophilic myelocyte. Nucleuses are still round in shape. And then gradually amount of these granules increases. amount of granules increases inside, so respective to the name of this myelocyte, and nucleuses became bean-shaped. And changing of shape of nucleus is reflected in a name as metamyelocyte. Metamyelocytes. Later, this bean-shaped nucleus became more elongated. It forms a bent neutrophil. And this bent neutrophil later forms the neutrophils. Bent eosinophils form eosinophil, and bent basophils form basophil. Okay, that's the main idea. Finally, we can talk about monocyte formation. Monocyte formation is started from same common myeloid progenitor, or a common myeloid progenitor gives rise to colony-forming unit granulocyte and monocytes, and granulocytes and monocytes gives rise to formation of monocyte progenitor. Then monocyte progenitor maturation is very short. So monocyte progenitor, so it doesn't need to collect granules inside their cytoplasm. It needs to form only lysosomes in the cytoplasm. Yeah, that's why maturation of monocyte is the shortest event. And from the round nucleus, it forms a bean-shaped nucleus. So here it's shown a little wrong, but actually bean-shaped is like this, bean-shaped. And monocyte with these features will extrude into the bloodstream, and it's already matured monocyte. Later in connective tissue, we'll discuss when monocyte entered into the connective tissue, same monocyte can produce macrophages. So, but it is a point of discussion in connective tissue. Then finally, we reach the point of lymphocyte formation. Lymphocyte formation starts from common lymphoid progenitor, CFUL, and it gives rise to three lineages of blood cells: is pre-NK cells, pre-T cells, and pre-B cells. But potentially, this common lymphoid progenitor gives rise to prodendritic cell formation, so which later forms the dendritic cells, and we will discuss it in next semester in a topic of lymphatic system. In a topic of lymphatic system, we will discuss what is dendritic cell function. But now we will concentrate our attention only on these three cells. So pre-NK cells, they are produced in bone marrow, and they are matured in bone marrow and entered into the bloodstream. And but keep in mind that when they formed in bone marrow, later their final maturation goes in their respective organs which are responsible for maturation of the cells. And NK cells and B cells are matured inside the lymphatic nodules or lymph nodes, in lymphatic nodules and in lymph nodes. But T cells maturation goes in a thymus. This final maturation happens there, and later T cells are getting stored inside so they stored in a blood, and also they're stored in lymphatic organs. In lymphatic organs like spleen, like lymph nodes or lymph nodules. So then in NK cells, T cells, and B cells are matured in this way. NK cells and B cells are matured once again in lymph nodes. T cells are matured in thymus. But later thymus will be degraded. Thymus is active until the puberty. But after the puberty, thymus is gradually shrinks and gradually disappears. And that's why on the population of T cells which were collected before, they will form the T cell population of the human body. Okay, let's talk about this arrow. Yeah, so it's shown that B cell is matured into plasma cell. So here we didn't discuss it in previous lecture. B cells actually they have a potency to be activized into the plasma cell. Plasma cell is so take a look. Inside the plasma cell we have a lot of ribosomes. So that's why it's blue in color, so pink and blue in color. And these ribosomes are able to produce antibodies. So actually, activation of B cell forms the plasma cell, and plasma cell is producing the antibodies. Antibodies which help us in our immunity response. So, and plasma cells are not present inside the blood; they are present only in connective tissue. So I hope you got it. So if I'm not mistaken, that's it. Yes, that's it. So, so how much time is now? We have a few minutes. I'll show you general explanation. By monophyletic theory of hematopoiesis, so hematopoietic stem cell is appeared inside the blood islands in the blood islands in the yolk sac, then gradually it enters into the bloodstream of embryo, then they settled in the liver, then from the liver it reside into the bone marrow, and in bone marrow hematopoietic stem cells are divided into two cell lineages: common lymphoid progenitors and common myeloid progenitor. Common lymphoid progenitor is responsible for three cell lineage formation: NK cells, T cells, and B cells, which in histological review, by histologically they are absolutely similar, and we can't find the difference between the cell morphologically. We can find it only with help of special stains. And then another lineage is myeloid progenitor. Myeloid progenitor gives rise to granulocyte monocyte progenitor and megakaryocyte erythrocyte progenitor. Granulocyte monocyte progenitor gradually developed into the 4 or 5 cell lineages: neutrophil progenitor, basophil progenitor, eosinophil progenitor, and monocyte progenitor. And also potentially it is responsible for prodendritic cell, but it's not point of our discussion. Actually, this we'll discuss it in next semester, as I said before. And granulocyte monocyte progenitor produce granulocytes, neutrophils, basophils, and eosinophils, and monocytes also. And MEP, megakaryocyte erythrocyte progenitor, gives rise to megakaryocyte progenitor cell and erythrocyte progenitor cell. Megakaryocyte progenitor cell, by the process of mitosis, forms a megakaryoblast. Megakaryoblast, by the process of endomitosis, forms a megakaryocyte, this giant cell, which produce platelets by splitting of, by extrusion of some parts of cytoplasm into the bloodstream. But erythrocyte progenitor, by the sequence of events, they start to produce more ribosomes. Then ribosomes produce hemoglobin. Then nucleus is shrinked and then extruded. And after this forms the reticulocyte, and reticulocyte entered into the bloodstream. And finally, final maturation of reticulocyte goes inside the bloodstream and forms the erythrocyte. It was explanation of entire topic in 2 minutes or 3 minutes. So if you listen my lecture carefully, adequately, so you could understand this lecture at all. So on this main part of my lecture is over, so I'll check the attendance again. So now only 80 students are present. So now checking. So this lecture video will be saved in YouTube. So then I can check your attendance every single day. So now let's save your chat. Okay, I saved your chat. Okay, and now if you have any questions, you can ask me. Okay, and now if you have any questions, you can ask me.
Sir, what is a mast
good
cell?
question. So mast cell is a. So let me show it to you. Mast cell is a cell which is very common by function. It's very common to basophils, and in previous lecture a little said about this. Mast cells are so by progeny it's similar to basophils, and they are present only in connective tissues, and their function is a promotion of inflammation. Inside the mast cells, we have same granules as in basophils, and with histamine and bradykinin and the heparan sulfate and all of this stuff. And due to some kind of irritation by antigens, these vesicles released outside, and it promotes the inflammation reaction. So in more details, inflammation will be discussed in pathology, but in connective tissue topic, I'll also touch this topic, touch this point in the next lecture. Okay, but main point you understood, yes, that mast cell is a very close relative of basophil, which facilitate the same function, permit the same function. Okay, any more questions? Yes.
Sir? Sir, how will you take the practice class of blood
Separately,
and
so we have
blood
one
formation
class of
together
blood
or
and one class of
separately?
blood formation. Any more
Okay.
questions?
No, sir. Sir, can you please mark my attendance?
Okay. If you are here, so I marked your attendance. So no more questions? Okay,
No, sir.
thank you.
No,
I
sir.
hope it was
No,
informative,
sir.
and I'll upload this lecture into the YouTube so you can repeat some points. So you see, just by reading the book and reading the lecture notes, it will be almost impossible to learn this topic. So very hard topic, and I do understand this. That's why my request to you, if you confuse in some points, watch this video again in YouTube. Okay, thank you for attention. See you next time.